As Martina said, my name is Niklas Ekman. I work for the Finnish Medicine Agency as a senior researcher, so a CMC or quality assessor. I'm also a member of the Biosimilar Working Party and alternate member of the Biologicals Working Party. And on top of that, I'm the rapporteur for this revision of the quality guideline. So what I thought to do is here now, I hope that you are all familiar with the revised guideline that we have had out for quite some time already. So I thought just to bring some of the issues that we have considered during the revision. First, a very short history. As you probably know, the first guideline, quality guideline, was adopted already in 2006, so it has been out for quite a long time, and we have gained experience since that a lot. So the revision work started in 2011, and the first draft was released for consultation in 2012. Approximately 1 year ago, we had an end of consultation and received a quite large number of comments. So we had from 14 stakeholders approximately 100 pages of comments. This is what we are currently working on. We are we have a quite good situation at the moment. We have gone quite far. However, there are some topics still that we felt as a drafting group that we need your input on. So I'm going to try to highlight these things and then hope for a good discussion after that. So a new thing that was introduced into the revised quality guideline was the quality target product profile. And first of all, I would like to quote the ICH-Q8 guideline to say what actually QTPP is. It's a prospective summary of the quality characteristics of a drug product that ideally will be achieved. I would like to point here the two words, prospective and ideally. So in other words, it's not, first of all, it's not a static thing. The QTPP might evolve during the development. However, it should be established at an early stage, and it should be used to guide the development of the biosimilar. So in other words, it's a development tool, not anything else in this sense. Related to the QTPP is the discussion on the expression system, because obviously in order to achieve the QTPP, you need to be very careful and consider the expression system that you are using for your biosimilar. Knowing that novel expression systems, or even different, may introduce additional risks, for example, atypical glycosylation patterns. In the draft guideline, we are also acknowledging the fact that actually both the biosimilar and the reference will have its own life cycle. So with regard to the biosimilars, the changes that are introduced should during the manufacture or developmental phase should clearly be covered by the ICH Q5E guideline. So this guideline does not cover that. These changes and comparability exercises should be addressed separately from the comparability exercise versus the reference, so the comparability for biosimilarity. However, it's always very advisable to generate the required data at all levels using the commercial or the manufacturing process meant to be the final one. Because otherwise, the situation can get very difficult if you have different comparability exercise on top of each other. Two things that we have also clarified in the guideline is, first of all, the formulation. It does not need to be identical with the reference product. And the second thing is with regard to the stability and the shelf life. So the shelf life of the biosimilar should be determined according to the ICH Q5C guideline. To say it clear, we are not accepting extrapolation of shelf life from the reference to the biosimilar. And to the comparability exercise, what we are asking for is an extensive usually side-by-side comparability exercise, where you assess the composition, physical properties, primary and high-order structures, purity, product-related isoforms and impurities, as well as the biological activity. Just to make it clear, this has always been the case, but now it's spelled out also in the guideline that the target amino acid sequence should be confirmed, and it's expected to be the same as the reference product. Product and product-related substances present in the finished product of the biosimilar should be highly similar to that of the reference medicinal product. And it's clear also that if you have big differences there, it might be difficult to claim biosimilarity. For the comparability exercise, we ask you to come up with target acceptance criteria. Perhaps it would be better to call it comparability ranges for the quality attributes. These can be based on the ranges used in the QTPP. So the range is defined during the process development. However, there might be a situation where some refinements are needed at the end levels. However, the limits should be quantitative whenever possible, taking into account both the number of reference medicinal products lots tested, as well as other things also, such as the quality attribute investigated and the test methods used, so the variability of the test methods. It's important to realize that this limit should never be wider than the range of variability seen in the reference product. Then what you very often hear at meetings is people mixing up the ranges for comparability with the release limits and acceptance limits, but these are two things that we would prefer to keep separate. I was previously talking about the life cycle, so we have also introduced some wording on the life cycle of the reference product. So during the development of the biosimilar, it do happen that the reference product evolved and the quality profile is changing. So this could lead to development of a biosimilar which is not according to a QTPP, which is or according to the profile of the reference product at that point on the market. Usually this is, however, acceptable, so we can accept both the pre and the post change ranges for the quality attributes. Then one new section that we have put into the guideline is the section for monoclonal antibodies and related substances, where we recommend to compare the affinity to the intended target, binding affinity to FC related and related other receptors, as well as the ability to induce both Fab and FC associated effector functions. We are aware that this is very much overlapping with the non-clinical section. However, we would like also to see at least a summary in the quality part of this. So as said, the deadline for commenting of the document or the guideline, draft guideline, was already approximately 1 year ago. We got a lot of comments. Here I have listed some of the main comments. So first of all, a lot of discussion around the QTPP. Then we got or a request to give further clarity on the use of different and novel expression systems. We also had a lot of comments dealing with the terminology, so whether comparability should be used or biosimilarity in inter or intra comparability. Clarification requested on appropriate use of public available standards. Obviously on the global development, the thing that we were discussing in the first session. Then also comments regarding the acceptable differences that can be seen on quality side. This is a very tough question, I don't think we're going to be able to put into a guideline detailed information on this or recommendation. Then we also had some issues with regard to the CTD requirements, so where to put everything this into the CTD. However, I thought that we should concentrate on some of these things. So where we got the most comments were perhaps the QTPP issue, as well as the use of different expression systems.